ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯ ಕಡೆಯಿಂದ ಈ ಸಂದರ್ಭದಲ್ಲಿ ಬಂದಿರುವ ಎಲ್ಲರಿಗೂ ಸ್ವಾಗತ. ಗುಡ್ ಮಾರ್ನಿಂಗ್, ಗ್ರೀಟಿಂಗ್ಸ್ ಫ್ರಮ್ Karnataka Science and Technology Academy. Welcome everyone to this series of Nobel Prize awardees lecture series 2021. We have with us our esteemed guest this morning, Dr. Bhupesh Mehta, Professor from Department of Biophysics, NIMHANS, Bangalore. Esteemed members of the Academy, Professor B.G. Munimani and Professor S.K. Saidapur. My colleagues from KSTA, Dr. M. Ramesh, CEO, the coordinator, Shri Umesh Datye, and all the participants. To all of you, a warm welcome. As you are aware, last 3 days, we have been having this Nobel Prize awardees lecture series. We had the physics and chemistry subjects yesterday, day 4. And today we have Dr. Bhupesh Mehta, as I said, from NIMHANS, speaking on Nobel Prize 2021 in the field of medicine or physiology. Dr. Bhupesh Mehta, Masters from Kumail University, PhD from NIMHANS, Bangalore, postdoc at the Yale University in USA, a number of, and also for a long time at the Yale, specialized in synaptics, the epilepsy, and related issues, and at Yale for a long time, and also at NIMHANS now, with a number of publications and research projects. With this, I have great pleasure in thanking Dr. Bhupesh Mehta for accepting our request, and also welcome you to this talk on Nobel Prize in Medicine Physiology 2021, Dr. Mehta. Over to you, Dr. Mehta, please.
Thank you, Dr. Ayappan, for the kind introduction. So I'll start with the slides. Good morning, one and all. Today I'll give you an insight on the 2021 Nobel Prize in Physiology. I'll just share my slides. I hope it is visible to all.
Yes, please go ahead.
Yeah.
Sir, please go
So today I'll give you an insight on the 2021 Nobel Prize in Physiology or Medicine, which was shared by Dr. David Julius and Dr. Adam Pataponian. So for a second, just let's think for a while, how do we know our world? So the first is by sight or vision, the second is by hearing, the third is by taste, or the fourth is by smell, and by feeling, or you can say touch or temperature, correct? So ample work over the years have been done on vision, how the photoreceptors sense light, the process of phototransduction leading to opening of cyclic GMP channels and neurotransmitter release, thereby relaying the transmission to the ganglion cells and from the ganglion to the visual cortex. Similarly, for hearing, the auditory system, where the sound wave causes mechanical deformation of the auditory hair cells towards the kinocilium, thereby opening the potassium channel and thereby again leading to neurotransmission. And this response is seen in the higher end of the brain. So similarly, with respect to gustation or taste, with the discovery of the taste buds having taste pores, here are the taste pores, the taste hair, and information being sent to the cerebral cortex via the synapses in the brainstem and the thalamus. When we smell the odorant gets dissolved in the mucus. The cilia of the olfactory bulb takes the odorant information by opening or closing the cyclic nucleotide gated channels, leading to neurotransmission to the olfactory cortex. So much has been talked or has been learned about the taste, the olfaction, the the sense of vision as well as auditory, hearing, but not much is known about somatosensation till the work of the Nobel laureates. So this is very old theory of René Descartes. In 1664, in the Treatise of Man, René Descartes theorized that the body was more similar to a machine, and that pain was a disturbance that passed down along the nerve fiber until the disturbance reaches the brain. His theory stated that it is the mind that senses, not the body. So this is a very recent image of a sensory nerve ending on the skin. You can see the green fluorescent protein showing the nerve endings. So external environment stimuli such as heat and pressure activate ion channel receptors in the membrane of the sensory nerve, resulting in electrical impulse which travels by the dorsal root ganglia. Here are the dorsal root ganglia to the sensory region of the brain. So this year, the Nobel Committee member, Sir Patrick, informed Professor of Tissue Biology of Karolinska Institute, announced the Nobel Prize winner for Physiology and Medicine. What he stated in his speech was, "You can feel the warmth of the sun, the coolness of the morning dew, a caressing summer breeze, and the fine texture of blades of grass underneath your feet. These impressions of temperature, touch, and movement are feelings relying on somatosensation." This is an illustration from the Nobel Committee for Physiology or Medicine. And the illustrator was Matthias Kerlin. So these are the awardees for the 2021 Nobel Prize in Physiology or Medicine: David Julius and Dr. Adam Pataponian. So Dr. David Julius is from the University of California, San Francisco, and Dr. Adam Pataponian from the Scripps Research in La Jolla, California. So before we move on to their work, let me give you a small introduction of the eukaryotic cell. If we see a eukaryotic cell has a plasma membrane, which makes up a boundary to avoid the mixing of the cytosolic component with the extracellular substances. So thus it keeps the cytosolic component far away from the extracellular world, and it is just the lipid bilayer, the phospholipid bilayer, with sphingolipids, cholesterols, and as well as proteins that separates out the two components, the cytosol from the extracellular space. Looking into an actual world, when we look the inside to the outside of the cell, the ionic composition of inside is very variable from the extracellular. It has its own importance. We'll be seeing it in the next few slides. When we look at just the sodium concentration inside the cell, the sodium concentration is very less in case of eukaryotic cell inside the in the cytosol, whereas in the extracellular environment, the sodium concentration is very high. It's in the range of 130 to 160 millimolar, whereas inside it is 5 to 20 millimolar. Looking into the potassium concentration, it is vice versa. Inside the cell, the potassium concentration is very high, and outside it is relatively low, in low 4 to 8 millimolar range. Similarly for calcium, which is a very important signaling molecule when it comes to cellular signaling, inside a cell, the calcium concentration is very low, and higher calcium concentration is considered to be toxic. Whereas in the extracellular surface, the calcium concentration is around 1.2 to 1.4 millimolar. So now if we see an electrochemical gradient has already happened because of the ionic disturbance or ionic distribution outside to inside of the cell. So if there is an electrochemical gradient, and this distribution of charges inside to outside of the cell has made a membrane potential difference between the two sides. So this is what the Nobel laureates also in their experiments exploited out. By putting an electrode inside the cell and an electrode outside the cell, they could monitor the membrane potential of a cell. And by using an amplifier attached to it, they could change up the membrane potential by tweaking on the voltages through the amplifier. This is the patch clamp setup that was used, that these laureates used for their experiments to find out, for their findings. So a patch clamp setup has an amplifier, it has a microscope, it has micro manipulators so that you can take small pipette or electrode near to the cell, an electrode holder, there's a stage, micromanipulator controller, because precise movement in submicron level is not possible through manual control, stage controller, and here is a glass capillary which has been heated and made into a glass micropipette. The tip of the capillary is in submicron level, is in the size of submicron, and here we have patched a hippocampal neuron with the same micropipette with a tip diameter of less than 1 micron. If we see this cartoon, at the tip, the micropipette is touching the cell, and we have taken the membrane inside the micropipette. Do remember, inside the glass capillary or the micropipette, the composition of the solution or the internal solution is similar to what is in the cytosol, with high potassium concentration, low sodium concentration, almost negligible calcium. And with this cartoon, we can understand that if one electrode is inside the micropipette and the other electrode is in the bath or the extracellular surface, we can make potential changes to the membrane. Thereby, we can induce currents. We can also record movement of ions through ion channels which are present in the cells. So the 2021 Nobel Prize in Physiology, as I said, was shared by Dr. David Julius and Dr. Adam Pataponian. So David Julius was born in 1955 in New York, USA. He received his PhD in 1984 from University of California, Berkeley, and was a postdoctoral fellow at Columbia University in New York. David Julius was recruited to the University of California, San Francisco in 1989, where he is now a professor and chair of Department of Physiology, and is a Morris Herson Chair in Molecular Biology and Medicine in UC San Francisco, California. So over these years, he has won several awards, out of which the most prestigious Nobel Prize in Physiology or Medicine was awarded to him in 2021, the Cowley Award for Neuroscience in 2020, Breakthrough Prize in Life Science 2019, and several other awards. So the Nobel Prize was credited for his discovery of TRPV1, or the temperature sensing channel, as well as the cold sensing Trpm8 channel or the receptors. So now coming to his work, Hot Science of Chili Pepper. He, in his study to figure it out, what exactly the hot chili pepper does. Why exactly we sweat when we take up chili, or temperature. Why we feel the heat when we take or rub capsaicin in our hands. So what he did is he took the sensory neuron, the dorsal root ganglion neuron, and he isolated the complete all the RNAs from it, mRNA, and made a cDNA library by converting our mRNA to cDNA fragments. And out of this, he got 2.4 million cDNA fragments. Now, this 2.4 million cDNA fragments, he expressed them in Neuro2 cells. So, and then started isolating that which fragment caused a change in physiology when exposed to capsaicin or the chili. So this was the study, the first paper from his group giving insight into the presence of capsaicin sensing receptors. So these are the clones respond. These are the 2.4 million cDNA fragments. He divided into 20 into 24. Sorry, just I'll So he divided into 24 pools, and out of that, the first one is just the normal pcDNA3 that he took, and he subjected it, exposed it to capsaicin. And these are the HEAC293 cells. The normal pcDNA3 cells did not respond to capsaicin, the pcDNA3 expressing cells. Whereas from pool DRG11, he observed that few of the cells responded to capsaicin. These are the bright greenish yellow cells that responded to it. He further subdivided this pool into individual cDNA and then found that the VR1 cDNA, when expressed in HEAC293 cells, it responded to capsaicin. This is how, by screening up 2.4 million clones, he found out that the one single clone, the VR1 or the vanilloid receptor 1 expressing HEAC293 cells, responded to capsaicin. And in the next step, he used capsaicin and stimulated the DRG neurons with capsaicin, and also observed an invert current. So as I told earlier, if you have an electrode inside a neuron and an electrode outside, any change or any opening of voltage gate or any opening of a channel will cause the influx of ions, like against their as per their concentration gradient. Like sodium, as a sodium channel opens up, a huge amount of sodium gets influx from 140 millimolar to inside the cell where the sodium concentration is relatively less. He observed that a cationic current was observed, a huge influx of ions was there when he exposed it with capsaicin or the VR1 or the vanilloid receptor agonist resin ferrate toxin. In the next one, he used capsaicin and then used the capsaicin receptor blocker or the vanilloid receptor blocker, and he observed that the capsaicin induced current was gone. So thus he inferred that the DRG neurons have capsaicin or the temperature sensitive receptors. In the next one, he took pepper extracts to see if the VR1 or the vanilloid receptor 1 responded to it, and also looked into the selectivity of these ion channels. He observed that when he stimulated with capsaicin, he saw an invert current. And same with the ball pepper also, the habanero, he observed an invert current. Thai green gave a smaller current, wax gave minimal, and Poblano Verde, another chili, did not respond or did not give any response or capsaicin response in the cells expressing VR1. In the next step, he looked into capsaicin response in absence of calcium outside, thereby indicating that whether the capsaicin stimulating receptors or the VR1 receptors are only allowing passage of the cation calcium, or they are allowing other ions also. He observed that in absence of extracellular calcium, still capsaicin was able to induce an invert current, and that current was consistent upon repeated stimulation. Whereas in case of calcium, the first response was there, and then slowly there was a decrease in the calcium influx. So to further test out the presence of VR1 in mice, he took, because these experiments were mostly in cultures or in acutely or isolated DRGs, so he next tested it in VR1 null mutant mice. The image shows IHC staining of lumbar spinal cord section from wild type and VR1 null mutant mice. We can see that the VR1 is completely, the expression is lost in the null mutant mice. He also looked into the expression pattern and saw that the knockout animal or the VR1 null mutant animal did not have the VR1 expression. So upon stimulation with capsaicin, in case of the wild type, a response was observed, and same with the vanilloid receptor agonist also. Whereas in the mutant, there was no capsaicin response or even the vanilloid response. Later, he found out that there's by using the behavioral paradigm like the Von Frey trail pressure, tail immersion, temperature or hot plate, radiant paw heating, he observed that whenever an animal, a mutant animal, when exposed to high heat, it was unable to respond as quickly as the wild type mutant. So if you see the tail immersion test, it is much more like control. This is the control. And in the mutant, the response was like more or less like near to normal. Whereas in case of the wild type, the reflexes of the animal were very, very fast. And same was observed with the hot plate test, where we put the animal's paws on a hot plate and see its ability to remove it within a set duration of time. In case of wild type, the ability was very, very fast, the latency was very fast, whereas in case of the mutant, the null mutant, the latency was low, thereby indicating that this VR1 channels or the TRPV1 channels are the heat sensing channels. Similar experiments in CHO cells were also performed by our second Nobel laureate also in case of the cold sensing receptors. So now moving on to his second discovery, or TRPM8 channels or receptors which are activated by menthol and cold. He took excitable cells, the DRG cells, and then exposed them to cold. He changed the buffer to 14 to 16 degree temperature and observed that few of the cells responded to cold. And same with menthol also, few of the neurons responded by fluorescence imaging. You could see a change in the calcium influx inside the cell. These experiments were, all the fluorescence experiments were done with the calcium sensitive dye Fura2, which goes inside the cell, and upon the catching up of calcium, any influx from the extracellular space, it fluoresces. So upon capsaicin stimulation also, he could see a quite many number of DRG neurons getting stimulated. By this, he could infer that there are receptors that in dorsal root ganglia, there are receptors that do get activated when subjected or when exposed to menthol or cold. And these receptors were called as Trpm8 receptors. And this is the paper in which he reported about the Trpm8 receptor. Identification of a cold receptor reveals a general role for TRP channels in thermosensation. In another sets of electrophysiology experiment, what he did is he exposed at two different potential of minus 80 and plus 80 millivolt. He exposed it to menthol, and he observed a current immediately upon exposure to menthol. And same when he used cyclohexanol, he was unable to see a structure similar to menthol. And when he changed the temperature, like usually all physiology data are done at 37 degree, he changed the temperature to below 20 degree, and still in cold he could see a response of the cells. And similarly with menthol also. So similar experiments at the same time were also done by Adler, where he used CHO cells to see the expression of TRPM8 channel and the sensitivity to cold. Now about our second Nobel laureate, Adam Pataponian. He shared the 2021 Nobel Prize in Physiology or Medicine. He was born in 1967 in Beirut, Lebanon. In his youth, he moved from a war-torn Beirut to Los Angeles, USA, and received a PhD in 1996 from Caltech, California Institute of Technology. And he was a postdoctoral fellow at the University of California, San Francisco since 2000. He is a scientist at Scripps Research, La Jolla, California, where he is now a professor. He is also a Howard Hughes Medical Institute investigator since 2014. Of the several awards that he has received, the major ones is the topmost is the Nobel Prize. Then he also received the BBVA Foundation Frontier of Knowledge Award and the Kavli Award for Neuroscience, Rosenthal Award of Distinguished Work in Basic Medical Research, and Young Investigator Award from the Society of Neuroscience. And this Nobel Prize was created for his discovery of the piezo 1 and the piezo 2 channels and the TRPM8 channel. These are the pressure channels or the pressure receptors. So his study is also very, very interesting, where he took 71 candidate genes and did expression of these 71 candidate genes in a cell line. So he observed that Neuro2A cells were the best cells to take into consideration because they showed response upon mechanical force. So he cherry picked out 71 genes, and these 71 genes he started knocking off each and every gene in the Neuro2A cells by using siRNAs. The 72 candidate genes encoding possible mechanoreceptor protein gene. One of the 72 genes was new and entirely unknown, mechanosensitive ion channel, and he named it piezo1 after the Greek word for pressure. Later, piezo2 was also discovered by his own group. So, upon mechanical force, the piezo1 and the piezo2 open up and they allow ion influx inside the cell. So, instead of a capsaicin or any other agonist acting on the receptor, here the mechanical force itself is good enough to open and close the ion channel, which he coined it as piezo1 and piezo2. So now coming to his study, where he looked into several cell lines and chose upon the Nero2A1 cell line, which was the best to show mechanically activated or current. He called it as MA current. So this is the paper, the first paper describing about piezo1 and piezo2. So the mechanical stimulation was done on a Nero2A cells, which was whole cell voltage clamp, patch clamp, and then with every micron extra stimulation of the Nero2A cells, he could see an invert current in the cell. Now, looking into a single channel activity, he observed that at every poke, some current was observed or some isolated, entry of influx was observed. And this is also another experiment where he used pressure. He calibrated it out in terms of mercury pressure, and then observe that for the duration of the pressure on the cell, the channel opening was observed, and this channel opening was very small, around 5 picoamp of current which he could observe. So this is all the members that he took. And then out of these 72, he could see only one. The 71s are the negative ones which did not respond to pressure. Only one of them responded to pressure. So he took up that candidate gene, the piezo1 gene, and used a scramble peptide which was mimicking it but did not had all the peptides same. And also on the other hand, he used an siRNA by knocking off piezo1, and he observed that in the scramble or in the control Nero2A cell, upon mechanical stimulation, a current was observed, whereas in the knockdown in the siRNA treated cell, the current was gone upon mechanical stimulation, thereby confirming that piezo1 is responsible for the mechanically for the influx of ions upon mechanical stimulation in Nero2. Then he looked into different cell lines, including the Higgs cells, and overexpressed it. What he observed is that Higgs cells do not express the embryonic kidney cell, human embryonic kidney cell, do not express piezo1. But upon overexpression, he observed that even the Higgs cell also, Hig293 cell also, upon application of pressure, showed an invert current. So this was in the same paper in Science. So the same experiment at different time point with different pressure, like in mercury, scrambled as well as in siRNA, he observed that piezo1, when it is not being expressed in the cell, did not showed any invert current. So the next one was piezo dependent large mechanically stimulated current is kinetically distinct from piezo1 induced current. In this one, he observed that in N2A as well as in Hig293 cells, when he was comparing, he saw the response was much more broader in case of piezo2 dependent, and it was much more relatively a longer current that he observed in piezo2, even though there was huge similarity in both the genes, but the current that they induced upon mechanical stimulation was different. So now, as we know, as on today, the sense, the temperature, the cold stimuli, as well as the touch that we feel is because of the hard work of these two scientists who extensively did a laborious work, like taking into account 2.4 million candidate genes, looking into 72 genes, and then figuring it out that this is the TRP1 channel which responds to temperature, this is the Trpm8 channel which responds to cold, this is a piezo or a touch proprioception channel which has a role to play in our sense or our feel that we get. And for this, a core body temperature, inflammatory pain, neuropathic pain, visceral pain, protective reflexes, mechanical pain, urination, respiration, how our lungs work, blood pressure, almost each and every place these channels are involved. And this knowledge of the TRP1, TRP8, and piezo channel is being used to develop treatments for a wide range of diseases, conditions, including chronic pain. If we just look into the citation history of these two scientists, every year their group is getting like more than like 3,000 to 4,000 citations. That tells us like how important that finding is for basic research, like from even from the translation point also. Thank you very much, Dr. Ayappa.
Thank you, Dr. Mehta, for this talk, bringing out all the details of the theme for which the Nobel Prize in Medicine Physiology was awarded for the year 2021 to these two illustrious scientists. You have given us the entire background of both the theme as well as the researchers. Thank you very much. We're grateful to you. At this time, if there are any comments, suggestions, you're welcome. From the audience, we request if there are any comments, remarks at this stage, please. From the participants, please. Any comments?
As of now, there are no questions from the
Yeah.
attendees.
Hello.
Hello.
Professor Mulimani is there.
Sir.
Yeah.
In the paper, there are two Indian authors.
Yes, sir.
Yeah, I mean, from where are they? And
Sir, I did not had a look at like their like I mean to say their status, like current status, where exactly these people are working, like whether they are scientists or into industries, sir. I didn't had a look.
yeah, Professor. Another thing is, this distinction, hot and cold you are making. Is it not possible that continuous temperature change?
Correct,
Why
sir.
two separate things?
No, like when we talk about supposedly taste buds.
Yeah.
So for taste bud also, like you would be very much surprised that for each and every taste, there is a separate bud, and never a single bud will detect both like two tastes at the same time. So same is with here also, like there are receptors that are only detecting cold temperature. There are receptors that are detecting. That is why they are characterized into TRPV8, that is for cold, and then TRPM8 is for cold, and then TRPV1 is for hot or chilly.
No, I was just a range of temperatures, where you define cold, hot, and is it a continuous thing, say up to 16 or something, you say 20.
Correct. So most of the experiments out here, because these were not performed in humans, these were in rats as well in cultures.
Okay.
In cultures, we have seen like if you take the temperature down, our observation also. So the cells do not last for very long. So it is basically because like these channels getting opened up, like they have done it at 16 or 18 degree.
Oh.
If they bring it down, because these channels are very non-specific, and like outside, as I mentioned, calcium is very high. As I mentioned, outside sodium is very high. If a huge amount of calcium enters inside the cell, it causes cell death, because a huge amount of calcium also is not very good. So, but the subcharacterization, I do not know at what all temperatures they had done these experiments. I believe like it was very laborious.
Thank you, Professor. Thank you very much.
Thank you. Thank you very much. If there are no other remarks, we would like to thank Dr. Mehta
Sir.
from NIMH for
Sir,
this.
may I ask a question, sir? I'm Dr. Kushal Das from BLD
Yeah,
University.
Dr. Kushal Das, please.
Yes.
Yes, sir, thank you, sir. How are you, sir?
Thank you, Dr. Sir.
Thank you. It's an extremely good presentation. I heard it, and I'm very happy to see my mentor, Professor B.G. Mulimuni, sir, is also here. And Saidapur sir also, I saw that he was here. Your talk was excellent, and you so nicely and so briefly, but to the point, you have explained the works of the two greatest Nobel laureates in this year. And I will say that they deserve, actually the work deserve Nobel Prize long back, because I'm basically a physiologist, and I'm a medical physiologist and vascular physiologist by research. And the work always fascinating when we teach the student about the sensory receptors and sensory pathways, specifically for pain, temperature, touch, three receptors we actually discuss here. Nobel laureates are mainly the temperature and pain. So these receptors, when we discuss, we are feel actually that how they have perceived the whole idea and analyzed through the brain. And here, as you mentioned correctly, that the part of the brain actually ultimately the main function. I have only one curiosity, that is the receptor point of views, that sensory point ending when it goes, because pain sensation are having the free nerve endings and other not. So there are two different, that's called C fibers. So they're different way they actually take the sensations, and when it goes to the sensory cortex, and there it implies, and the stimulation that is quite different. Do you think that the role of the chlorine, which is actually not been pointed out, the chloride, which is a very much negative by nature and very low in concentration extracellular fluid, made an influence to sensations at the type of this stimulation point at the pain or temperature and the perception point at the cerebral cortex? According to that, whether these two, you have mentioned about the calcium, okay, they have also said about the calcium, you explained. Do you really think, because you were a scientist, do you think that there is a role of the chloride actually not discussed and not been said? It is not you, it is, they have not mentioned about the chloride roles here very vividly. By blocking the chloride, by patch clamp when they're using, they can block the chloride also to movements and see the other ionic movement in the both the side. They did it, the calcium and other ions. But what about the chloride? That is ignored, and which is a very important for threshold, specifically in the cardiac diseases when the heart pain occurs. That is a signal for pain sensation, and that leads to the ischemic heart diseases and many of the phenomena. So role of the chloride was very vital for the cardiac disease. Do you have any ideas that why they skip the chloride part here? So that's my question.
Sir, there are, I completely agree with you. There are some limitations of the patch clamp experiments also, because I have been doing it like routinely for almost 15, 16 years. So what happens is, when we look into the ionic composition, as I said in the earlier time point, like most of the like ion channels that have been talked about, like the calcium, voltage gated calcium channels, the voltage gated sodium channels, even the voltage gated potassium channels and all, they are all cationic. So with respect to chloride, like chloride has been talked mostly about like when it acts as an exchanger. So there are exchanges which take up the chloride and all. As such, like the trigger point, like whenever the trigger point is there, whether it is mechanosensation or inotropic response, when it comes to the first influx is always like a high concentration sodium or calcium getting inside. So very less has been talked about, like when we talk about like in actual physiology, about chloride being the first trigger, like showing a change or a rush from outside to inside. Like if we talk about sodium chloride, the sodium chloride outside the cell is 140 millimolar, and inside the potassium chloride is around 140, 120 millimolar. So if you see in actuality, there's not a very much big difference in the chloride concentration in comparison to when we talk about sodium and calcium. So the first will be like, even as a chemist also we talk about like a diffusion, like a free diffusion from high concentration to low. So it will always be against the, it will always be as per the gradient. Like if 140 sodium is high inside, outside, then it first thing that will rush inside will be sodium, because it has to cover up the full cytosol to make it almost equivalent. So that is why probably chloride, like it's not like of course in heart like it has a big role to play,
Yes.
but in the sensory system as well as in the retina and all, not much has been explored in that area, sir.
Thank you. Thank you very much for excellent explanation. Thank you.
Thank you, Dr. Das. Thank you, Dr. Mehta. Very nice having you with us. Grateful to you. Now over to Dr. M. Ramesh, CEO of the Academy, for his words at this stage.
Good morning. We have listened to a very insightful and informative talk by Dr. Bhupesh Mehta, Associate Professor Nimmans, on research by Nobel laureates in physiology and medicine this year. Dr. Mehta gave a glimpse of their studies. Dr. Julius, David Julius, University of California, who utilized capsaicin from chili peppers that induce a burning sensation to identify a sensor in the nerve ending of the skin that respond to heat. Then Dr. Arden from Scripps Research Institute, California, who used pressure sensitive cells to discover a novel class of sensors that respond to mechanical stimuli in the skin and the internal organs. On behalf of KSTA and all the participants, Dr. Mehta, thank you very much for this scholarly talk. Thank you very much.
Thank you, sir.
I am also grateful to our Honorable Chairman, Professor S. Ayappan sir, for encouraging us to organize Nobel Lecture Series this year too. Last year it was organized well. This year again it is a very successful one. I am also thankful to the presence of Professor B.G. Mulimani sir and Professor Saidapur sir, our esteemed KSTA members. Thank you very much, sir. I am also grateful to Professor Kushal Das sir for his presence, and senior academicians Dr. Ganesh Raj, Professor C.D. Patil, Dr. Abhishek, Dr. Mukunda, Dr. Devraj, Dr. Venkat Lakshmi, Dr. Purnima, Dr. Akshata, and Dr. Manu Kumar also have joined this talk. Thank you very much, all of you. And I also thank all the participants who have joined this talk on Cisco WebEx and YouTube platform. I wish you all a great day. Thank you very much. I would also like to extend my sincere thanks to Dr. Rangnath sir, Honorable Member of KSTA, who initiated this Nobel Lecture Series program last year. Thank you very much, everybody.
Thank you. Thank you very much. Thank you. Thank you, Dr. Mehta and everybody. Thank you.
Thank you, sir. Thank you.
Dr. Mehta, thank you very much.
Thank you very much, sir. Thank you very much.
Dr. Bharat suggested your name. In fact, he was supposed to come for this talk also. Since he has some urgent meeting, he could not
Correct, sir.
attend. He has conveyed best wishes to you. And
I'll convey, sir. And
Thank
sir, thank you very much, sir. Thank you.